We just need a couple more scans, and then you should be ready to go.
It seems like we've done a good job getting through this treatment, and you've been very brave through this all.
Thank you so much. This proteomics treatment has done wonders compared to any of the chemotherapy sessions that I've been to before. I wish more people were able to experience this aside from me. You guys should totally pitch this to a group of entrepreneurs.
That's actually not a bad idea. However, it's very costly and therefore we would need some funding. But what do you think about the idea?
I'm totally in for that. Let's work on getting this patient discharged and then discuss it later. But it's a great idea.
So what do you guys have to pitch to us today?
We believe we have come up with a way to cure cancer.
Are you sure about that? Many people come to us with false prototypes and they end up failing.
We have tested this technology on a patient last year. In fact, we actually prepared a video that we can show to you guys so that you would be able to better understand how the technology itself works.
So let's see how it works.
What exactly is cancer? Cancer refers to any one of a large number of diseases characterized by the development of abnormal cells that divide uncontrollably and have the ability to infiltrate and destroy normal body tissue. Cancer often has the ability to spread throughout your body. More than 11 million people are diagnosed with cancer every year. It is estimated that there will be 16 million new cases every year by 2020.
Some background information on proteomics and how it can cure cancer. The term proteome was first coined in 1994 by an Australian doctor named Mark Wilkins. His definition of proteomics refers to the total set of proteins expressed in a given cell at a given time and protein-protein interactions.
Proteins are the building blocks of the living organism made of different amino acids. They vary greatly in their function, stability, three-dimensional structures, and are considered as the final gene products of most genes. The target of drugs are typically proteins, which is the things that do the work in your body. Your protein come from your DNA, but they're not made directly from your DNA. They're made through RNA, which instead instructs your cell to make proteins.
The collection of proteins is called proteome, and for the first time through proteomics we can really see the thousands of proteins at once. It is using a sophisticated computer software to break the sample down so that every protein present is identified by analyzing its three-dimensional structure and its amino acid sequence. The two major techniques used in proteomics is electrophoresis and mass spectrometry. Electrophoresis, which is the separation of complex protein mixtures, and mass spectrometry, which is an identification tool that follows thousands of proteins in your blood. You can analyze the proteins in people's blood, and the reason for the blood is the garbage can of your body. So when you get sick, cells will often die or viruses will cause problems of things and it will end up shedding diagnostic proteins in your blood.
So, with the use of proteomics, researchers often study people's blood to see if they can see some unusual proteins and to see if something is wrong with a person. The goal is to discover new proteins that are called biomarkers. They reflect if something is wrong with a person's health. Ideally, we like to have markers for every disease you can imagine to be able to follow that in a very simple blood test. For instance, when men get prostate cancer, they often make a particular protein called PSA that gets elevated, and due to that, cancer cells have more protein than there would be normally present. So, in summary, proteomics is an amazing field where you can follow biomarkers and learn all kinds of changes in people's bodies, which lead to getting the right drug to the right person.
So how can cancer actually be cured through proteomics? One major development to come from the study of human genes and proteins has been the identification of potential new drugs for the treatment of disease. This relies on the genome and proteome information to identify proteins associated with a disease, which computer software can use as targets for new drugs. Cancer can be treated through this technology by use of blood out of a patient, and this blood out of a patient is used by proteomics to sort out the proteins based on weight, slipperiness, and size. The 3D structure of the protein allows for differentiating the different features. This is done by overlaying the samples to identify what is different and similar about a human with the disease and without the disease. A molecule that fits the active site of an enzyme but cannot be released by the enzyme inactivates the enzyme. This is the basis of new drug discovery tools, which aim to find new drugs to inactivate proteins involved in diseases. As genetic differences among individuals are found, researchers expect to use these techniques to develop personalized drugs and are more effective for the individual.
Until recently, cancer was generally treated with radiation and chemotherapy. However, these methods also destroy healthy cells. Proteomics is being used as a diagnostic tool, and early data from the projects have been very positive, with the computer software managing to identify 100% of ovarian cancer samples and 96% of prostate cancer samples. With these positive results, it is surely only a matter of time before diagnostic proteomics is seen in a clinical setting.
Both the technology and pharmaceutical arenas will be highly impacted by the new window proteomics is opening. This area of studies will permit us to identify, characterize, and quantify proteins on a massive scale, resulting in a fundamental change in biological and chemical approaches to understanding proteins and even the ability to address new important questions. Advancing the tools of proteomic technology further is a major interest to biotech and pharmaceutical communities. In order to successfully develop protein drugs and effective combinations of pharmacological or biological inhibitors that can specifically target key classes of molecules, a thorough understanding of the combination of proteins dysregulated along a regulatory network is of major importance. Similarly, the determination of selected interactions between the proteins and other biomolecules, be it other proteins, nucleic acids, ligands. There will be more production of drugs in pharmacies, therefore increasing the sales of pharmacy as more customers will be buying and pharmacies will be selling to more industries.
Thank you for listening to our presentation, How to Cure Cancer Through the Technology of Proteomics. We hope you take this pitch into consideration. Thank you.